I'd like to begin by acknowledging the traditional owners and custodians of the land from which we meet today. WEHI is located on the lands of the Wurundjeri people of the Kulin Nation, and I'd like to acknowledge their ancient and continuing connection to land, sky and water. I'd like to pay my respects to elders past, present and emerging, and extend that respect to all First Nations people present today and at WEHI. So today we're going to hear from Sam Lee, his PhD completion seminar, and I have to preface my introduction by saying that sometimes I use the word interesting to mean interesting, and sometimes more than just interesting, but never uninteresting. And I would describe Sam's PhD journey as interesting. Sam commenced his PhD at the start of 2020, and you know what that happened in the 2 years that followed that. These were interesting times. As you'll hear, the theme of Sam's project was using biological knowledge and computational approaches to make sense of heterogeneity in different biomedical systems, in cell lines, in nanoparticle therapies, and in metastatic breast cancer, which are all interesting, fascinating areas. So I'll let you know what version of interesting I meant there. The breadth of applications that Sam's worked on really is reflected in the diverse group of co-supervisors that he's got here, and we're all here today. So Delphine Marino from Olivia Newton-John Cancer Centre, Matt Farrier from University of Melbourne, and me, and as well as his initial PhD supervisor, Melissa Davis. So fast forward a few years and Sam's journey got even more interesting. His initial supervisor moved to a new institute interstate and then left the country and left academia. So quite late he moved into my lab and I became his primary supervisor. That's why I'm doing the introductions. But something very cool happened, something that I'm really impressed by. It was interesting. Sam rounded up the three of us, the co-supervisors, and formed us into a supervision team. And I'd like to particularly acknowledge Delphine and Matt's really close involvement in supervising Sam to the end. And I'd like to acknowledge Sam's skill in herding the cats. It was really impressive. It was really also a delight to be part of. So over to you, Sam. I look forward to your talk, which I think will be very interesting.
Well, thank you everyone for coming today, and thanks Tony for that really kind introduction. That was really lovely. So today I'm going to be talking about the last 4 years of my PhD. So my thesis, which was looking at computational approaches for modelling heterogeneity across complex biological phenotypes, which is a bit of a mouthful, but really I've just tried throughout what I've been doing in my PhD is to look at how we can study biological systems with taking the really fundamental variation that's a part of them into account and really trying to make this the centre of how I've done the analyses that have formed my PhD. So biological heterogeneity is really all about the kind of fundamental variation that occurs across all scales in human systems, indeed all living systems, right the way down from like the molecules that enable metabolism to occur through to the kind of fundamental variation we see amongst human populations, and that's both in health and in disease. And so this biological variation, it really occurs within and kind of across all of these different facets and all of these scales, and they all really depend on each other. And I think it's really important, at least in my view, it's really important to consider this heterogeneity, especially in medical research, because it really underpins the kind of probably what drives complex diseases and what drives complex disease progression. So we kind of see this really prototypically in cancer, where it's all about the kind of changes that occur and how gene regulation and other processes occur to change differently. And so that's kind of really what I've tried to focus on throughout my PhD, and that's what I'll be taking you through today. So kind of there's three projects I want to talk about, and they tackle heterogeneity either between patients, so this idea of interpatient heterogeneity, or else I've also been looking specifically within the cancer context at intrapatient heterogeneity. So interpatient heterogeneity is all about the differences we see that kind of drives treatment resistance or changes in immune profiles. And also kind of this idea that clinical characteristics clearly have a really big role in how disease progresses, but we also kind of need to consider intrapatient variation, and this really starts with intratumoral heterogeneity. So this, which has been, I think, well established since the 1970s, probably even earlier, is that tumors are not a homogeneous group of cells. There's a lot of variation that occurs within them, both at the epigenetic and genetic level, and this is probably best captured these days with single-cell sequencing. This isn't actually a single-cell sequencing talk, but it's a really great way to think about it. And the way that I've really focused on this is an exploration of clonal dynamics. So this is the idea that different subgroups or different groups of cells within the tumor will behave differently and have differing fitnesses and metastatic potentials, and that kind of leads into this idea of intermetastatic heterogeneity, which is really the observation that metastatic tumors are frequently quite different to their originating primary tumor in their genetic and epigenetic background, as well as how they respond to therapies. And so especially in the context of breast cancer, trying to understand why metastatic tumors are much harder to treat is really critical, as it's still, so it's kind of metastasis is still what causes fatality for the vast majority of those who die with breast cancer. So that's a really important topic to study. And I'll be talking about these through three really quite distinct projects today. So first of all, I'll be talking about interpatient heterogeneity, looking at nanoparticle immune cell interactions and how we can try and predict this with the use of proteomics data to try and understand variation. And this is in a healthy human context that we're going to be looking at this variation. And then I also will be talking about how we can map patient tumors to preclinical models and try and do this at the individual level, as opposed to doing this at disease or disease subtype resolution. And then finally, I'll talk about work I've done more recently with Delphine Marino's group, looking at organization in metastatic breast cancer, organotropism, in metastatic breast cancer. Cool. So, three quite different parts of the talk, but we'll get into it. So this first part I'm talking about will be the computational analysis of nanoparticle immune interactions. And so this is work that I've done under Matt Farrier's supervision, and I'm really grateful for him for kind of years now of very patient advice and kind of feedback, which has been fantastic. So, nanoparticles, to give a very broad overview, they're tiny engineered particles that have really useful biophysical properties, and as a result are really appealing to use in drug targeting, especially for targeted therapies. And so we can functionalize these using either antibodies or ligands that we'd attach to the nanoparticle surface as a way to try and target where in the body they would go. And this functionalization and kind of the idea of targeting is really key to how we want to use nanoparticles in therapies. But what actually happens when we take our nanoparticle and we put it into human blood is this kind of thing called the biomolecular corona or protein corona, to be a bit more particular forms. And that's seen really nicely with this TEM microscopy image from this paper No et al. From a few years ago, where they're just showing uncoated gold nanoparticles on the left there, and then after incubation in serum, you can see they get this kind of halo or corona that forms around them. And it's this corona that's made up of both proteins and carbohydrates, as well as other macromolecules, that actually really forms what I'll call like the active surface of the nanoparticle, and immune cells, or indeed any other cells in the body, won't necessarily see and be able to respond to the nanoparticle itself, but to the proteins and other molecules that will be kind of absorbed to the outside. And work over the past 20 years or so has shown that really this protein corona is extraordinarily complex in its composition and is also dynamic. So it's a changing thing, and you can see that if you change the surface or you change the type of nanoparticle that you have, as a result, you'll get a different protein corona forming. So the way we measure these days, what we can do to measure the protein corona is using proteomics. And so this allows for the quantitative identification of both what proteins are attached or absorbed to the surface of our nanoparticle, as well as how much of them there are. So it is quantitative data, which is really important, and we typically LC-MS proteomics is used for this. So this kind of gives us a nice quantitative read of what exists on the protein corona. I think it's important to note here, this diagram makes it look extraordinarily simple. I think it's actually an extremely hard experimental task to actually isolate the proteins from the corona, which causes I will say difficulties in the analysis in terms of contamination. But it's also good to note, I think important to understand what protein corona data is in the world of proteomics and how it differs from most transcriptomics or indeed other kind of cell-based proteomics. So what I'm just showing here is a heat map of a nanoparticle proteomics data set from this paper, Zhu et al. From a few years ago. And what they did is they took this pegylated MS100 nanoparticle and incubated them in healthy donor blood from 23 different healthy donors. So this is 23 different patients, well, not patients, donors, and we've incubated the nanoparticles, and after that incubation, they have measured the protein corona, and that's been measured in triplicate, and what I'm showing here are the averages of those technical triplicates per sample. And in addition to measuring the protein corona profile, we've also got nanoparticle association of these nanoparticles post-incubation with those donors' immune cells. So specifically, they've got nanoparticle association with B cells and monocytes, and that's measured by flow cytometry. And I think what's probably pretty obvious when you look at this heat map is that it's exceptionally sparse. And what I mean by that is for many of these proteins, there is very little detection across most of the samples. And you see there's kind of this massive area in the middle, very low abundance, and this is after normalization. So we can see that it's really sparse data. There's also a massive dynamic range. So some of these proteins are detected at massive abundances compared to other ones, and indeed some of them are ubiquitously kind of expressed and detected, and some of them much less so. So it's quite complex data and has to be handled, I think, differently to regular kind of cell-based proteomics or transcriptomics. All right, and the question that I after looking at this data for a while, I started to wonder if we can actually predict immune cell association using protein corona profiles. And so what I wanted to do was to take the nanoparticle immune cell associations that had been measured using flow cytometry, and that's what I'm just showing here, are the mean fluorescence intensities for our nanoparticles for each donor. We can see a larger MFI for the y-axis for B cells, x-axis for monocytes, relates to a larger amount of association of that donor's nanoparticle profile with these immune cells. So we can see, possibly as expected, there's a pretty strong, actually, linear correlation between B cell and monocyte association. So I wanted to know, can we predict this? And the way I wanted to look at this, doing this, because we have this quantitative measurement of association, is using linear regression. So I looked at doing linear regression using lasso penalized models. And this is just fitting, so prediction of the association using the nanoparticle protein corona. And basically what I find is it's not predictive. And I've done this using a kind of cross-validation scheme where what I'm showing are test set accuracies. So I build a model, a linear regression model on 4/5, I kind of hold out part of the data, fit my model on that, and then on unseen samples that were not used to build the model, I test the accuracy of how well I can predict uptake. And what we see if we compare in this box plot, these red points here as kind of a model fit without any features, so it's a completely non-predictive model. We actually see that if I try and predict uptake independently per cell type or with a joint model per cell type, neither of them are actually significantly better than a random model, which was a bit disappointing, but I was maybe not super surprised because we don't have a lot of samples to work with here. So it's quite a constrained problem. But what I kind of wondered was, this was a very kind of simple, straightforward way to do it, and I wanted to know kind of if we could use our biological knowledge about how nanoparticles actually behave and would interact with immune cells to try and structure that analysis in a better way. And so I went back to the literature and I had a look, and I found this paper that got published that kind of really nicely indicates how we could think about nanoparticle immune cell interactions. And so in this paper, what they did was they did a genome-wide CRISPR knockout screen where they knocked out every protein coding gene independently in the U87MG cell line, and as the readout to their screen, they wanted to look at the uptake of gold nanoparticles as a result of knocking out that gene, of each gene in sequence. And kind of what they found was that the LDLR receptor was actually really critical for uptake of nanoparticles in this context. And they kind of went a little bit further. They could show that most of the secondary hits in their CRISPR screen were actually also downstream of LDLR, which really strongly implicated that specific pathway. And indeed, if they looked at the nanoparticle protein corona via mass spectrometry, they could see that relevant ligands for LDLR were indeed enriched on the nanoparticle surface compared to other ligands. So this was kind of really nice evidence that ligand receptor interactions could be used as a way to understand how nanoparticles and immune cells might interact. However, it was only done in a single type of nanoparticle and a single type of cell line. So it would be really nice for every nanoparticle that we're interested in if we could do a CRISPR knockout screen and identify the receptor of interest, but that's not feasible. So what I've tried to do is use our knowledge of ligand receptor interactions to actually build a new way of understanding and scoring our data set. Cool. So what I wanted to do was to predict nanoparticle cell association using ligand receptor scores. So instead of trying to predict nanoparticle cell association using protein corona abundances, I'm instead going to predict nanoparticle cell associations using a ligand receptor score that combines both our nanoparticle and our cell type information. And so to build a model for nanoparticle cell association, there are a few different things that are required. I've kind of already gone through how we've got nanoparticle cell association data, and so this is the measured flow cytometry associations of our immune cells with the nanoparticles, and we've already looked at the protein corona abundance, which is the experimental data of interest. But if we want to integrate ligand receptor information into this, we'll obviously need some sort of way of knowing what ligands and what receptors might be interacting. And for this, I turn to ligand receptor databases, which have really come out of the single cell analysis field in the past 5 or so years, especially with cell-cell interaction, where a lot of work has gone into looking at what ligand and receptor interactions might occur. So there are now really nice public databases, including OmniPath, with a lot of information for this. And what we'll also really critically need to make this cell type specific is an understanding for different cell types of what receptors are being expressed on them. And I don't just want binary information of yes, this cell type is expressed, no, yes, this receptor is expressed on the cell or not. I actually want quantitative information to be able to say how much of this receptor is expressed. So I'm going to go through on the next slide how I've kind of generated this using site-seq data. And then once we've got our cell type specific receptor profiles as well as ligand receptor databases, we can then use this to generate scores of ligand receptor interactions per nanoparticle cell pair. And so to generate the cell type specific receptor profiles, I turn to using public site-seq datasets, as these are probably the best way currently to measure cell surface abundance of receptors, as opposed to trying to measure either the gene expression of genes that are responsible for receptors or intracellular protein expression and then trying to extrapolate out to the cell surface. We can just directly measure what proteins are on the surface and how much of them there are. And so this quite a large CiteSeq dataset was published a few years ago, where they looked at 218 different proteins on the cell surface from about 48,000 PBMCs from healthy donors. So this gives us a really nice big reference, and it's also well annotated. We already know for all the different cells, whether they are B cells, CD4 cells, CD8, dendritic cells, monocytes, or NK cells, and so that gives us a really nice truth set, which we can then use to build cell type specific receptor profiles. And what I had to do in order to make these suitable for scoring is I really needed to denoise the data a lot, because without kind of very specific normalizations, there's a really high background expression in CiteSeq data. That means scores won't be sparse, and they will not be they will not be sparse enough to be able to score with in a system. And so by using the DSB method, I was able to generate sparse profiles per cell type. I can then average across cell types to get this profile of cell type receptor expression, which is just what I'm showing in the heat map here on the right. And we can see that, as expected, each cell type has a pretty unique range of receptors that it is expressing at quite different amounts. So this is then what I can use to kind of integrate cell type specific information into my modeling framework. And so once I've got these cell type specific scores, I can integrate them with the nanoparticle protein corona dataset to generate nanoparticle cell scores using ligand receptor information. And as part of a package I've been developing, I've got this function here that will generate those. And once we've got those scores, here I'm just showing a principal components analysis of those scores. Hopefully that will disappear. And we can see that kind of as expected, there is a nice strong cell type specific structure in these. And as a kind of validation of these, I wanted to look at how these individual ligand receptor scores would correlate with cellular association across donors. And so again, I'm just showing the associations for B cells and monocytes on the left, and on the right, I'm showing for a selection of these ligand receptor scores that had significant correlations against both the monocytes and B cells. And then I'm just showing the correlation coefficients on the X and Y axis respectively. And what's really nice to see here, and is a really good kind of positive control, is that we're seeing the kind of complement interactions, so C3-CR1 and C4a-CR1, so these ligand receptor interactions being really positively associated with cellular association of these nanoparticles. So we're seeing that if a nanoparticle has a higher complement interaction score via either of these interactions, it is going to be associating with immune cells more, which, given kind of how the complement system works for detecting foreign particles in blood, that makes a lot of sense. So it's a really nice positive, good positive control that these scores are meaningful. And so what I wanted to do, what I wanted to do now was to model nanoparticle cell association using these ligand receptor scores instead of just using the nanoparticle protein abundances. And so I went through the same kind of cross-validation scheme to fit models using lasso regression. Unfortunately, after quite a lot of modeling, they still have quite poor predictive ability. And so what I'm showing here are kind of the residuals plots after modeling. And what we can see is the observed, so the kind of the known association on the x-axis, and then the difference between the predicted association and the true association on the y-axis. In an ideal world, all of these points would cluster kind of randomly and nicely around the 0 line, which clearly they're not. This is quite a biased model, and we can see that it's really under-predicting at both extremes, which indicates that the model probably doesn't have enough information to be able to fit appropriately in a kind of machine learning test train framework. but I had a look at the model coefficients just to kind of see if it made any sense at all, and I compared them to kind of known opsonins and disopsonins in the literature. So opsonins being particles that will promote interaction with immune cells, and disopsonins those that will reduce associations with immune cells. And again, it's nice to see we're getting the complement interactions coming through with positive model weights, and also we're getting some known, this HRG, which is a strongly known disopsonin for nanoparticles specifically, we're seeing that it is getting a negative weight. So it seems like these are not models with predictive accuracy, but maybe there's something going on. I then tried a lot of different things to improve these models. I looked at whether I could model them using categorical associations, so just saying for this cell type, it's got high or low uptake, as opposed to trying to predict the exact amount of uptake. I tried to model it using binary scores, so just saying, yes, this interaction occurs in this nanoparticle cell association, or no. And I also tried different normalization approaches to see if changing how we're dealing with the data before modeling would make a difference. Unfortunately, none of these were able to have significant differences. However, I then kind of wanted to take a step back and tell if there was any explanatory value of this data modeling approach at all. So what I did is I kind of did some permutation testing. And so before generating my nanoparticle cell scores using the cell type specific receptor expression, what I did is I randomized the cell type specific receptor expression so that that contained no structure. And what I'm showing here are just two MDS plots, the top one of scores generated without randomization and the bottom one with randomization. And I think what's really clear to see, the top one, these scores have a nice structure that kind of pull out the different groups within our data, but the bottom one has no structure at all. And then, so what I did was I fit linear regression models using the true data or using the permuted data. And so that was either by randomizing just the cell type data or randomizing both the nanoparticle and cell type data. And this time, instead of doing test train splits, I fit it on all the models and then just looked at the AIC, which is the information criterion that kind of captures the amount of information or the explanatory value of a model. And because these are fit on all the data, they're not predictive models, but it gives us a lot better sample size to work with to hopefully be able to see if there's any signal at all. And what's really nice to see with this formulation of the AIC score, I'm using a larger AIC, means a more explanatory model. We see that when we permute the model, we're losing information in a stepwise fashion. So we can see that the model fit with real data has the largest AIC, which is nice. And then if we permute just the cell type data, we lose a lot of information from the model, but we lose even more information if we permute both the nanoparticle and cell data as expected, because this model effectively has no information now. But the fact that the model with permuted cell type data has more information is a nice way of showing that indeed the cell type information is adding information into this model. Yeah, cool. So kind of as part of this project, what I've hopefully kind of gone through and looked at is how interpatient variability in a healthy context has a really strong effect on immune cell recognition of nanoparticles. And I've focused here on talking through how I've built a new framework for ligand receptor focused analysis of nanoparticle protein corona data, which it cannot accurately predict association of nanoparticles with immune cells yet. And I've looked at this in a couple of different data sets and results are pretty similar, but we kind of do consistently see that incorporating the cell type information does improve the explanatory performance of models if we're not fitting them in a predictive framework. And so also as part of this work, I've been trying to generate an R package to really try and standardize how protein corona analysis can be done, as it is still, it's a very new field. So it's something that I think has a lot of potential, but the kind of standards of analysis just need a little bit of work still. So now I want to move on to kind of talking about the work that really started my PhD, And that was looking at how we can use preclinical models to understand patient tumor data, and this will be in the context of using gene expression, so RNA-seq data sets. Cool. So what I'm just showing here are a couple of principal components, analysis plots of a large collection of cell lines, so the Cancer Cell Line Encyclopedia on the left here, which is around 1,000 different cell lines for all major types of cancer, as well as the Cancer Genome Atlas, which is a large compendium of patient data, primarily of primary tumors, for, again, all major types of cancer. And I think what's really important to take away, and when I look at this, what I see, is we see clustering within, I've colored them by disease type here, we see clustering within disease types, but there is also massive heterogeneity and differences within each disease type. So we kind of see really large intermixed complexity when we look at these data sets at a kind of a really high scale. And so for me, this really kind of raised the question, if we have a specific patient tumor, how do we actually know what the most similar cell line is using gene expression data? Because we can kind of imagine that if you wanted to take a patient tumor from an individual's patient tumor, given the large kind of heterogeneity within each cell type, so within different diseases for each cell type, it's hard to say which one would be the most appropriate. And so why we might care about mapping patients to preclinical models is really this idea that different models for the same disease, we kind of see that they can respond very differently to treatments. So we know that if we take 5 different cell lines for any disease and treat them with the same drug, we're probably going to see a variety of responses. And we also know that if we take a number of patients and we treat them in the same way, we're going to see a variety of responses. So what would be really nice is for those patients, if we could actually tell which of those cell lines was going to be the most appropriate, and so we could hopefully be able to decide before treating patients possibly which drugs will be more effective, or indeed which drugs might not be that effective. And so this is really kind of the question I started with, and I really wanted to look at how we could do this using gene expression data. But this question of how we can measure sample similarity between data sets is a really confounded and quite a difficult question for technical reasons. And so obviously when we're comparing between data sets, batch effects and gene expression data have been really shown to be critical and change. If we compare 2 data sets without taking batch effects into account, it's really hard to understand the similarity between them. And there are obviously also quite significant biological differences between patients and preclinical models that we need to somehow better take into account or be able to ignore in a smart way, perhaps, to actually be able to compare patients to preclinical models. And what I found when I started looking at the literature back in 2020 was that basically all of the approaches for people that had tried to compare preclinical models to patient tumors to date at that point had relied on batch correction, which makes a lot of sense for explanatory analyses where you want to take 2 cohorts and put them together and try and understand them. But in the case where you have only a single patient or a handful of patients, you can't actually do batch correction. It's not possible to batch correct in that situation. So none of these methods were actually appropriate. So I wanted to kind of ask the question of, is it possible to do this without batch correction? And really the hardest part of that is knowing how we can benchmark it and how we can measure our benchmark. And so in order to actually do this benchmark of matching samples between different data sets, what I did is I turned to using kind of proxy data sets. And this is because if we took a data set of cell lines in patients, there's actually no way to truly know which patient should look the most similar to which cell line. But if we take multiple cell line data sets that have the same cell lines and have each had gene expression done on them, and here it's really important that these are multiple data sets that are high quality and we know are using good quality cell lines, we can compare between them. And we should be able to say that if we take, say, an MCF7 cell line from one data set and compare it to a second data set with a whole bunch of cell lines, including MCF7, we want whatever method we use to say MCF7 looks the most like MCF7. And so by using a data set with a lot of different cell lines, we can then calculate accuracy of how well we can match. And so that's just very simply, it's the number of correct matches from our query data set into our reference data set, and then just divided by the number of query samples we've actually looked at. And so in this talk, I'm going to be using results from 3 different cell line data sets. So we've got the NCI60 data set, a cell line atlas from Genentech, which has around 500 cell lines, and also the Cancer Cell Line Encyclopedia, which I've previously mentioned, which has results from around 1,000 cell lines. And so what I'm just going to show here quickly is kind of what these results should look like. And so what I wanted to compare with different normalization approaches, different feature spaces, so what genes we look at, as well as different dissimilarity measures to see which combination of these would give us the best matching performance, which is here on the y-axis. And when I looked at this, we kind of looked at 8 or 9 different distance measures, multiple different normalizations, and a lot of different feature spaces. So the number of results becomes very large, but what's really critical to see is that Spearman's dissimilarity here that is consistently the highest performing and is always giving us, if not, it's always giving the best quality results, and nothing else ever does quite as well. So what's really nice, so this is good evidence kind of that using rank-based methods is a very effective way to match between data sets. And obviously, because most of the other methods use batch correction, I wanted to compare to see what effect batch correction would have. Here, I'm just showing the Genentech and NCI60 dataset on the left, pre-correction, on the right, post-correction, and what we can see, so they correct really nicely. Here, again, they're all just colored by disease type, so we can see that I've managed to batch correct these 2 datasets quite nicely together. And then I wanted to compare to see whether batch correction would make our matching better or worse. So what I'm showing here, first of all, is on the y-axis of these 2 plots, it's kind of the matching performance using our batch-corrected data, and on the x-axis, it's matching performance using our non-batch-corrected data sets. And we can see that here, so L2, this is the Euclidean distance, which is a very standard distance measure. It gets better results using batch-corrected data than non-batch-corrected data, but Spearman's dissimilarity actually starts to get worse results when we start to use non-corrected data, which was quite a surprise to me. I then compared whether using the Euclidean distance on batch-corrected data was better or worse than using Spearman's on non-batch-corrected data, and I was actually really pleased to see that using Spearman's on non-batch-corrected data is still actually better than using batch-corrected data, which is a really nice way to kind of see the power of using quite simple methods like Spearman's dissimilarity for these problems. So just to wrap this section up quickly, what we can really see is that using cell lines as both the testing and training sets allows us to do quantitative benchmarking, and that Spearman's dissimilarity is a really effective and robust method for matching samples. I haven't shown it here, but I also looked at this work comparing PDX models to primary tumors they were derived from and found very similar results in that using Spearman's dissimilarity is very effective for matching patient tumors to PDX models as well. And so now I just want to talk about the last part of my project. And so this is work that's been done in collaboration with Delphine Marino's lab at the Olivia Newton-John, where her and her team have really welcomed me in the last 9 or so months, which has been a fantastic opportunity. And I've been very grateful for it. And there's going to be a lot of data in this part of the talk that has been generated from a lot of work from John and Srija especially. So I'm very thankful for them for that. And it's been really fun. Cool. So here I want to focus a bit more on this question of intra-patient heterogeneity, and especially this idea of how intratumoral heterogeneity can lead to intermetastatic heterogeneity. So what we see within a tumor is that we'll have specific or different clonal populations that might that often will have genetic and epigenetic variation between them and will show different clonal dynamics. And by clonal dynamics, what I mean is that we'll see that some clones will become dominant over other clones over time, and indeed, it's only typically a small proportion of clones, so common groups of cells within a tumor, that are actually able to form metastases. And so what we see when metastases form is often they are quite different to the original primary tumor, and trying to understand why this is a really important question, and especially in the case of cancers like breast cancer, we see very different rates of where metastases occur, and also mortality is very dependent on where metastases occur. So understanding what drives different metastatic locations to be able to grow is a really important question. And this is a question that Delphine's group has been asking for a while now, and using clonal barcoding methods, they're really able to look at this in a fantastic way. So what I'm showing here is a Lego barcoded tumor from a mouse. So it's a human tumor grown in a mouse, and we can see the different clonal populations via their fluorescent coloring. So each of these small dots is a different cell, and we can see the kind of complex relationship of how these different clones interact with each other. And so this is just a really nice kind of way to see how complex the intratumoral heterogeneity of a individual tumor is. But what's really nice about this Lego barcoding method is we can do a lot more than just look at our tumors with it. So the way it works is we take our kind of unlabeled population of cells, and we take lentiviral particles that contain these fluorescent barcodes, and they can then be added to the cells, and then selection done to be able to get a group of a set of barcoded cells where each clone, so each group of individual cells within that tumor, are able to be labeled with a distinct and unique set of fluorescent tags, which we can then use to identify them. We can grow these tumors in mice and then wait for metastases to form. And then after both the primary tumors and metastatic tumors have been resected, we can analyze these tumors using microscopy and also sort them into pure clonal populations using flow cytometry. And it's this ability to then be able to sort out the individual clones from both primary and metastatic tumors that lets us look at some really interesting questions, because we can then go on and do bulk or single cell sequencing along with many other kind of downstream analyses. Cool. So the work I'm going to be showing here is kind of just a small part of this larger organotropism project we've been looking at using Lego barcoding. And so in this, we've been studying metastatic tropism to the brain environment, to the lungs and the liver, using orthotopically injected mouse models. So we've got both metastatic and non-metastatic clones within the primary tumors we're looking at, so we can compare metastatic competence using these. And we've also done this using multiple different models, including MDA, MB231 derived tumors, and a number of different patient-derived PDX models. And we can also validate this in patient cohorts. So what I'm just going to show here is some single-cell sequencing data of Lego-tagged MDA MB231 cells that have been grown in vitro and then sequenced using single-cell sequencing, and then clustered into this UMAP here. And so this is work that was done by Jean Berthollet and Andreas. A manuscript is currently in preparation for it. And so what's really interesting about this UMAP, although it kind of looks like any other UMAP on first glance, is that actually the clusters are not colored on the basis of gene expression, but on the basis of those Lego barcodes. So we actually know that for each cell, we know which clonal population it's a part of. And then what's really nice to see is actually each of these different clonal populations has a unique gene expression signature that we can kind of see forming in the way that the gene expression clusters actually represent independent clones. There's obviously a bit of mixing in the middle, but it's pretty good. And what's really nice to see is that actually we know in this model, clone 6, 16, and clone 4 are kind of the most aggressive clones So these are the ones that metastasize more frequently and often will be dominant in a tumor in terms of the proportion of cells in the tumor. We can see that these are actually the most unique and kind of separate the most from the other cells. So it's really nice to see that the more aggressive clonal populations actually have more distinct gene expression profiles, which is really nice. Of course, this is just in vitro, so we wanted to look at it in vivo. And what's really important to understand about this system when we grow these MDA MB231 cells in vivo is we see really strong differential organotropism. And what I mean by that is I'm just showing the proportions of clones 16 and 6 here for simplicity, but these typically do form most of the tumor anyway, is that we see that in the tumor you get, this is an average across 9 mice, we're seeing about 75% of the tumor is clone 6, and the remaining is clone 16. Then if we look at liver tumors in this model, we see that it's almost all clone 6. But if we look at the lungs, we see clone 16 coming through more strongly. And we can actually quantify this a bit more robustly if we compare the ratios of clone 6 and 16 in each of these sites. And here I'm just showing results across 9 different mice. So we can see actually there are really strong differences in kind of the clonal fitness between the tumor, liver, and lungs, which is a really kind of interesting phenotype to have seen. So we wanted to follow this up by doing some RNA sequencing. And so what I'm showing here is we've done bulk RNA sequencing of paired primary and metastatic tumors from 3 different mice in this cohort. So we sequenced, in addition to the primary tumor, we've got lung, liver, and lymph node metastases, and in each of these sites in each mouse, we've also sequenced both clone 6 and clone 16. So 24 samples in total, where each group, kind of each combination of clone and tissue is represented by three different samples. And we can see some really quite striking patterns emerge, and this is just the principal components analysis of all the protein coding genes. So this isn't just from selecting differentially expressed genes, this is just how strong these effects are. We can see that on the first principal component, we see really strong differences between clone 6 and clone 16. And then if we look at the second principal component, we can see it's really this lung environment that pulls out very differently. So we see that really actually interestingly in this model, we were seeing much smaller differences between the primary tumor and the liver and lymph node metastases than to these lung metastases. So obviously we did differential expression on this, and what we saw is that we saw really strong gene signature compared to lung metastases to primary, and we can do this as it's a factorial design using the main effects. So we're looking, these are genes that are consistently changing in both clone 6 and in clone 16. And we can see, we do differential expression analysis and then gene set enrichment analysis. We can see some quite strong signals coming through. And I just want to focus on one of them today, which is that we're seeing this upregulation of TNF-alpha by NF-kappa B in the lung metastatic environment compared to the primary. We also see this significantly enriched in the lung environment compared to the liver environment. And this was really nice as kind of like, because in previous work from Delphine's group, they've been able to show that TNF-alpha signaling was enriched in a different mouse model in the lungs as opposed to the liver. So it was really nice to be able to kind of confirm this result again, to see here the signal coming through. And also, given this experimental design, we can also look at differences between clones. So when we compare between clones 16 and 6, what was, we get here on the heat map on the left is the differentially expressed genes, so we get really clear differentiation of these clones. What's quite interesting is we actually again saw enrichment of TNF-alpha signaling. And so we saw that TNF-alpha signaling is upregulated in clone 16, which is the clone that we saw to be more dominant or more kind of growing out more strongly in the lung environment than it would in the liver environment, which was, pardon me, quite a nice kind of corollary. And we actually see this this enrichment of TNF-alpha signaling in clone 16, both in the lung environment and in the primary tumors. So it's starting to kind of get at this idea of how kind of variation in the primary tumor can also be seen in metastatic sites if we look at it from a kind of a clonal lens. So that was quite nice. And then to validate this, we wanted to look in another model. And so for this, we used a patient-derived xenograft model that was derived from a metastatic triple-negative breast cancer patient from the pleural effusion. Again, we barcoded this using LIGO barcoding, and then we could do in vivo experiments across multiple mice. And here, I'm just showing the PCA for that. We can kind of see, again, the lungs appear quite different to the primary tumors, which is really nice. And here we see strong clonal variation in the primary tumor, but we don't see it quite as much in the metastatic tumors, which is interesting. We can then do single sample scoring of these TNF-alpha genes using SING-score, and we kind of see similar patterns, which is really nice. The lung environment here, we're seeing higher scores than for the liver environment, and indeed, we're seeing here clone 12 to have higher TNF-alpha scoring than clone 4, and we know that clone 12 is the clone that is typically dominant in this model. So we can kind of see how this idea of dominance is holding across models, which is nice. We then finally wanted to look at this using some patient data, and so for this we could use patient autopsy samples from Brocade, which is a really fantastic rapid autopsy program run via the Peter MacCallum, where for patients who died with advanced metastatic breast cancer, they were able to do full autopsies and tissue collection immediately after death, and then make these available to researchers for developing models and as validation. And so what I'm just showing here is for the 9 Brocade patients that we've currently got access to, we've kind of got a mix of both ER-positive and triple-negative breast cancers, and also quite wide range of survival time. But for each of these patients, we've got multiple metastatic sites sequenced, and importantly, for 5 of them, we've got RNA sequencing of lung metastases in addition to other metastases. So here, what we can see is if we take these 5 patients and again score them using SING-score with the TNF-alpha score, we can see actually that TNF-alpha is enriched per patient. So these are the pink dots here, especially compared to the liver metastases for the patients that have both. So we can kind of see really nicely that this TNF-alpha signature being stronger in the lungs versus the liver is holding across multiple models and indeed in patient data. So it's really, yeah, I think nice to see how we can use these LIGO barcoded models and go right through to patient data. And that's just where I'm going to finish this part of the talk up. So how we can use bulk RNA-seq of flow-sorted populations so we can really study and ask questions, the intersection between clonality and organ adaptation. And in addition to this question of TNF-alpha, we're also looking at how clonal variation is driving brain metastatic competence and adaptation. I think there are some really interesting results we're starting to see there. And we can also look to see how well clonal identity is maintained over generations. And with that, I'll just wrap up quickly to say, this is, I guess, throughout my PhD, I've been looking at this question of how we can model both inter-patient and intra-patient heterogeneity in really quite diverse contexts and by using quite a broad array of methods. But I think I've learned an awful lot throughout this process, and I'm really thankful to all of my co-supervisors who've kind of led various parts of this project and of these projects, and I've learned a lot from all of them. So that just leaves me to thank everybody. There's a lot of people to thank, particularly my supervisors, Tony, Matthew, and Delphine, and also Melissa, who supervised the first part of my PhD. I really appreciate all the work you've put in throughout the years. It's been really fantastic. Everyone who was in Melissa Davis's lab, both here at WEHI and at Surgency in Adelaide, you've been all fantastic to work with, and it's been really enjoyable. And everyone as well in Delphine's group and the Tumor Progression and Heterogeneity Lab out at the ONJ. It's been really fun working with you guys, and I've learned an awful lot. And I also really want to thank my parents who have been, I believe, are listening online, so thanks, Mum and Dad. You've been really supportive. It's been really, really thankful. Jackson and Sarah, you've been really lovely to have here throughout these years. Ashley as well, wherever you're hiding. And especially Lucy, who you've really been fantastic. And I'm excited to be done with the PhD soon. Yeah, I'd be very happy to take questions. Thank you.
Have we got any questions in the auditorium or online? The co-supervisor's going to ask a question.
This is just to warm up the crowd, really. So I had a question about the PDX model, wonderfully presented, by the way. So on slide 40, you had a picture of the principal components of the metastases. Yeah, this one.
Yeah.
So the bottom left, and you said that there was less variation for the metastases in this case.
Yeah.
I'm wondering if you think, if you have a reason for this, or if you would like to give your opinion of my opinion about why that is. Yeah.
In this particular case, I think it's possibly to do that this batch of data has gone through, I think, one more passage in mice than previous data, so there's possibly we're actually seeing some selection of certain phenotypes within the clones that are going to be more aggressive in general because they're the ones growing out in the clones. So I think that's my take on why we're seeing less variation in the metastases.
So you think they're overall more aggressive?
Yes, yeah, I think, yeah.
Any other questions? There's one up there. We're almost precisely on time.
Thanks for the great talk, Sam. You really slayed that. Yeah, I might have missed this in your second talk. So you were trying to, I guess, set up a pipeline to compare what the best cancer cell line was to use for a particular, best cancer cell line that represented a particular type of tumor the best. And yeah, I guess with that TCGA data, did you ever end up comparing the cancer cell lines to that TCGA with your Spearman's dissimilarity score?
Yes, I did. And I cut that due to time constraints. But I actually saw some quite interesting stuff where so I took, including lung adenocarcinomas, and I compared them against the patient-derived models repository, which is a PDX database, which actually has very few lung adenocarcinoma models. And what I was finding was that these models, so these patients were really mapping mostly to pancreatic PDX models. And then when I dug into what that particular pancreatic adenocarcinoma model was, it was actually an extremely dedifferentiated pancreatic cancer that model was derived from, which kind of suggests that perhaps it had, there's an interesting link to cell type of origin stuff there. Yeah. But yeah, I certainly compared the primary tumors from TCGA against both cell lines and PDX models.
So also, I guess because those primary tumors are bulk RNA-seq, would you have to, deconvolute out, signatures from immune cells, or is that something that you had to factor in?
In this work, I didn't take purity into account, but I think generally it would be important to do, especially if you had, and I think I've seen this with a lot of the analysis I've been doing recently with the Brocade dataset, where we've got some samples that have beautiful sample quality, and then other ones where there is a lot more contamination from adjacent cells, immune cells. So you very much would need to consider purity. Yeah.
Okay, let's just thank Sam once again for a fantastic presentation.